Hi, it's Lemetria from Smart Edition Academy, and today we're going to go over the T7 practice test 1 for the science portion of the test. Before we get started, though, I wanted to let you guys know that you can follow along with this test by clicking the link in the description box where you'll have a free access to the entire T7 practice test. We have lots of practice questions, as well as having additional T7 resources such as the T7 study group, as well as the T7 online course. Okay, so you definitely want to follow along. It's going to take about an hour for us to go through each one of these 50 questions. We're going to go ahead and have, I'm going to go over each one and give you the reasons why it is or is not the correct answer. So you definitely don't want to miss this. So without further ado, let's get started. All right, you guys, so here's question number 1. Which blood group is a universal donor? A, B, AB, or O? So I'm going to go with O, and then we're going to discuss why. Here we have it. So with the blood type O, that is considered what is the universal donor out of the ABO blood groups. A person can be a universal donor or an acceptor. The universal blood donor has type A. A universal blood donor has type O blood, while a universal blood acceptor has type AB blood. So there are different types of blood groups that are a part of the major groups. You've got A, B, AB, and O. And then you've got the way that that's set up is based off of the antigens that are on the surface of the red blood cell, right? So with that, that's how they classify the blood. So if you look at this chart here, you've got your blood group A, B, AB, and O, and they can accept from all of these. But as you can see, type O blood can only accept type O blood. However, they can donate to everybody over here. So that's the one that you're going to want to choose if you happen to see this question on the test. And if you happen to see one that's talking about the universal acceptor, you'll know that that is type AB. So without further ado, we're going to move on to the next question. Question number 2. Blood oxygen levels are most likely low when blood leaves the aorta, fills the right atrium, reaches body tissues, or flows through the arteries. For this one, you guys, I'm going to go with fills the right atrium, and here's why. It says right here that blood continually flows in one direction, beginning in the heart and proceeding to the arteries, arterioles, which are smaller blood vessels, and the capillaries, which are even the tiniest of vessels that are all throughout our body. When blood reaches the capillaries, exchanges occur between the blood and the tissues. A lot of times it's gas exchanges and changes of different nutrients that we need. After this exchange happens, blood is then collected into the venules, which feeds into the veins and eventually flows back to the heart's atrium. The heart must relax between the 2 heartbeats for circulation to begin. 2 types of the circulatory processes occur in the body, which is going to be the systemic circulation, which is going to be where the left ventricle is going to be pumping blood throughout. And you're going to have the pulmonary circulation, which is the part where we're talking about right here, where you've got the 2 major veins, the superior vena cava, the inferior vena cava. That's going to bring the deoxygenated, which is the lowest oxygen content of blood, from the upper and the lower half of the body, right? So it's all going to converge together right there. So that's going to be the correct answer for this one. It's going to be when blood oxygen levels are at the lowest, thinking deoxygenated blood, you're automatically thinking the right atrium, okay? So there, that goes right there. And we're going to move on. Let's see. We have this cool little diagram right here, where it kind of shows the flow of the blood. So here's your right atrium right here. And that's blue, so that lets you know that that is going to be the deoxygenated blood. Whenever you see these pictures right here, you got the red, that's your oxygen-rich blood. The blue is going to be the deoxygenated blood. Okay, so there's the inferior vena cava, and then the superior right here at the top. It's all related together, and it's going to be dealing with the pulmonary part of the body. Okay, so let's move on to the next question. Question number 3 is an intracellular chemical signal can be produced in the cell membrane Once it is produced, where does it go? To a different cell, to the part of the same cell, to a region outside of the cell, or to an area of high ion concentration. They're trying to trick us up right there, right? Well, this takes us back to our prefixes right here. If you've had any of the medical terminology classes or anything, you may have heard about intra, inter, versus all that stuff. So right here, intra is going to mean within, together, and thinking along the lines of same, right? So right here, we're going to go with the part of to another part of the same cell. That's our keyword right there, same. So let's check our answer and see what it says. You've got intracellular receptors. These receptors are located in either the cytoplasm or the nucleus of the cell. Signals diffuse across the cell membrane and bind to the receptor sites on intracellular receptors of the same cell. Or you have membrane-bound receptors, which are receptors that extend across the cell membrane with their receptor sites on the outer surface of the cell membrane. They respond to intercellular chemical signals that are large, water-soluble molecules that do not diffuse across the cell membrane. Okay, so keep that in mind. When you see intra, automatically start thinking it's in the same area. Okay, so let's move on to question number 4. After food has been masticated in the oral cavity, where does it go next? That's a good question. Is it the colon, the liver, the pancreas, or the pharynx? So you've got to start thinking anatomy. When you hear a question like this, is it going to be the colon? Not so much, because we know that that's going to be in your lower gastrointestinal tract, your rectum, areas around there. The liver or the pancreas, not so much. Like that, the way you approach these questions is going to be you automatically are going to want to eliminate 2 questions immediately that you know are not the right answer. When you're taking these tests, which are going to prepare you for your bigger tests, like your NCLEX RN, you are going to have to eliminate immediately 2 choices that you just know aren't right. But these right here, where you could get tied up, could be the pharynx, or could it be the pancreas? So, some people may not know that, but the pancreas, they might think it's P, it's P, it's P. I'm going to go with pharynx, and here's why. Once the food has been masticated in the oral cavity or the mouth, it is then swallowed and travels back into the pharynx down into the esophagus, which leads to the stomach. So make sure you brush up on your A&P where it comes to the gastrointestinal tract, because you definitely want to know where things go once they hit the mouth, for sure. Next question. The diffusion of nutrients through the walls of the digestive system is critical to the homeostasis in the body. Where does the majority of this diffusion take place in the digestive system? Is it the stomach? Is it the esophagus, the oral cavity, or the small intestine? Now, when you hear the word diffusion, when you see these questions, try to pick out the keywords. Now, when you hear about nutrient absorption, it's tempting to say, oh, it could be the stomach or whatever. It could be, that's definitely where it could be happening. But no, that's not what this question is asking us. It's asking about the diffusion of nutrients. That, again, is going to take you back to your anatomy and physiology. And you can automatically say right away, of course, we know that it's not the oral cavity. It's not the esophagus. We know that those are the upper parts of the GI tract. So those we can eliminate right away. But you're going to be looking between the stomach and the small intestine. And I'm going to tell you right now, we're going to go with small intestine. And here's why. The duodenum is the first part of the small intestines. It's located between the stomach and the middle part of the small intestines, the jejunum. Once food has mixed with the stomach with the acid in the stomach, it moves to the duodenum, where it mixes with the bile from the gallbladder and digestive juices that are secreted by the pancreas. From there, in the duodenum, the absorption of vitamins, minerals, and nutrients begins. So it always takes place in the small intestine. Remember, the small intestine's always first. And as it goes along the tract, it's going to get more and more centralized into a solid stool. So it's going to have to go to the large intestine, which is where we get to the end product of digestion. So try and keep that in mind. Like I said, brush up on your AMP if this question kind of slipped you up between you were caught between stomach and small intestine. Definitely brush up on your AMP. Next question, you guys. As soon as an invader known as a blank enters the body begins to fight. Is it the antibody, pathogen, trigger, or vaccination? Now, we can automatically eliminate two of these, right? So I'm going to say straight up, I know it is not vaccination, you guys. That has nothing to do with the body fighting off things right away. That's actually something you induce to the body to help your body fight. But we also can say that trigger, that one doesn't sound sciencey enough for me, so I'm going to say, I'm not going to pick that one. But when you get to antibody and pathogen, that's where you might get a little bit tricky. Things might get a little bit tricky. But I'm going to tell you, pathogen is going to be the correct answer, and here is why. Let's see. So, a pathogen is an infectious foreign body that enters the body and causes disease or illness to the person. There are 5 types of pathogens. You have viruses, bacteria, fungi, protozoa, and worms. Pathogens have antigen proteins that are found on their surface that are unique to each pathogen. So right here it goes through, we've got antibodies, which are a protein that are produced by the body's immune system when it protects harmful substances, which are going to be the antigens. It's basically, this is the actual army coming to fight. The pathogen is the invader. The antibodies are the army. They're coming to fight. Vaccination, which is what I said, you introduce different types of pathogens and disable pathogens or a harmless microbe that are going to help your body's immune system to fight off potential infections. So with this particular one, it's going to be pathogen. So let's move right on to the next one. We have question 7. Where is skeletal muscle found? Keyword, skeletal, right? Is it inside the heart, attached to bone, lining the walls of the bladder, or within the gastrointestinal tract? I'm going to tell you guys, I'm going to go with, because of that keyword skeletal, I'm going to go with that is attached to bone. And here's why. Skeletal muscle is it's the long, striated, cylindrical type of muscle fibers. There are many nuclei in skeletal muscle cells, and they're attached to the bones in the body. Skeletal muscle contracts voluntarily, meaning you have to make it move. Keep that in mind. You have to make skeletal muscle move, and that is under conscious control. Whereas smooth muscles, the other muscles that this question is referring to, you've got cardiac muscle right here, and you have the smooth muscle right here. So this question is testing, do you know the difference between the different types of muscles? So smooth muscle is going to be that muscle that consists of non-striated muscle cells that are spindle-shaped. Like cardiac muscle cells, smooth muscle cells contain only one nucleus. That is a key differentiator. This muscle type is found in the walls of the intestinal organ, or in internal organs like the bladder and the stomach. And smooth muscle contraction is involuntary and controlled by the autonomic nervous system. Again, that's totally different from skeletal, which we control consciously. Cardiac muscle, I think you guys know, we don't control our cardiac muscles, right? That's going to be an involuntary muscle, and it's going to be totally separate. So right here, that particular cardiac muscle is going to consist of muscle cells that are striated, short, and branched. These cells contain only one nucleus and are branched and are rectangular. Cardiac muscle contraction is an involuntary process, which is why under the control of cardiac muscle contraction is an involuntary process and is under control of the autonomic nervous system, which is why I said we don't control our cardiac muscles. And this muscle is found strictly in the walls of the heart. So definitely, you guys, if you see skeletal muscle, It's going to be attached to the skeleton. Okay. All right, so let's move on to the next question. All right, so if a person smells something sweet, what form of information is this initially perceived as in the nervous system? Is it cognitive, integrative, motor, or sensory? We're going to go with sensory, and here is why. These are going to be the first initial, or I say the first, when you first notice it, it's going to happen in your brain first. So automatically when you see the word, what's going to be the initially perceived part in the nervous system, which is strictly referring to the brain and the spinal cord, it's going to be sensory. A sensory nerve is a nerve that carries sensory signals from the external environment to the brain to the central nervous system. It is also an afferent nerve, long dendrites of sensory neurons which send sensory information towards the central nervous system. It's sending information to the brain. This information is what is sensed using the 5 senses from the external environment, which are going to be sight, sound, taste, smell, and touch. Next question. Question number 9. While hiking, a person is startled after encountering a bear. Her palms get sweaty and her heart starts racing. Which part of her nervous system was directly stimulated? Was it the central nervous system? Was it the parasympathetic nervous system, somatic, or sympathetic? Which one are you guys thinking? This one's going to be a little bit tricky. So right away, I know we're going to eliminate central and somatic. And my 2 choices I'm left with are going to be parasympathetic or sympathetic nervous system. So you guys, we're going to go with sympathetic, and here is why. The autonomic nervous system is responsible for activities that are non-voluntary and under unconscious control. This system controls glands and the smooth muscles of the internal organs, heart rate, breathing, and digestion. The autonomic nervous system is further divided among the following, or into the following rather, which is going to be your sympathetic nervous system, which is what we just talked about. And that's going to be the sympathetic as in the fight or flight response. That's going to be triggered in times of stress or duress. Whereas your parasympathetic nervous system is going to control the involuntary processes that are unrelated to emergencies. So when you see parasympathetic, it's going to be dealing with your rest, digestion activities. Think right here, which I think is pretty creative and very helpful, which is going to be parasympathetic equals peace, sympathetic equals stress. Try to remember that if you guys see these 2 on a test in the future, which I'm sure you probably will. So let's go on to the next question, which is going to be, which of the following types of tissues include cells of the immune system and of the blood? Connective, epithelial, muscle, or neural? The 2 that I can get rid of immediately, you guys, are going to be muscle and neural. Muscle cells, we know, are dealing with muscle. Neural equals neurons, which equals the brain. That has nothing to do with the immune system or the blood. However, when it comes to connective or epithelial tissue, you may be in a little bit of a pickle right here if you're stuck between the 2. So I'm going to go ahead and go with connective tissue, and here is why I chose that one. The tissues that are connective are going to, let's just back up for a minute. A tissue is going to be a group of cells with similar structure and function and similar extracellular substances located between the cells. Let's take a look at this table right here to just brush up on the different types of tissues, which is why if you got stuck and end up picking epithelial, remember the epi, our prefix there, indicates on the surface, epi meaning outside, okay? So that's not going to be immune system or the blood. We know that those are strictly internal. However, connective tissue is characterized by extracellular material that separates cells from one another. The functions include enclosing and separating, connecting tissues to one another, supportive and moving, storing, cushioning and insulating, transportation or transporting, and protecting. So immediately right here we have an example that shows it's going to be cells of the immune system and of the blood, whereas epithelial is going to be on the surface, skin, and the linings of your internal organs. Okay, so definitely keep that in mind if you happen to see a question similar to this. Brush up on your anatomy where it comes to the different types of tissues. Next question. Which best describes homeostasis, a functional system of the body, blood flow to every cell in the body, a relatively constant environment within the body, or neural pathways that have integrated into the body? So, which are you guys thinking you can eliminate right away? Right here, I know I can say right here that homeostasis has nothing to do with the blood flow into every cell of the body. I'm going to get rid of that one. And I'm also going to get rid of homeostasis is not a functional actual system in the body. We know that that's not true. However, if you're stuck with neural pathways that have integrated into the body or relatively constant environment, I'm going to go with relatively constant environment, and here is why. Because we have that prefix right there that kind of clues us in. That's going to be homeostasis. When we see the word homeo, we're thinking constant state, homeostasis, right? So let's go ahead and check our answer, and it right here says that homeostasis is the existence and the maintenance of a relatively constant environment within the body. Each cell of the body is surrounded by a small amount of fluid, and the normal functions of each cell depend on the maintenance of its fluid environment within a narrow range of conditions. So it has to stay within that very, that area that it's the sweet spot, basically. That's the word for the sweet spot. That's homeostasis right there. So you guys, let's move on to the next question, which is question number 12. Fertilization, which is the fusing of one sperm and an ovum, results in a embryo, a fetus, an infant, or a zygote. Which one are you guys thinking? Right away, we can say that it's not an infant. An infant can be a child that's anywhere between 0 and 12 months. That's far too big to be just after the fertilization period. So you've got to think back to reproductive AMP right here, you guys. Is it going to be a fetus? No, because that's still a more advanced state of human. So we're going to nix that one out too, because a fetus, you can hear heart tones. You can feel the fetus moving. But is it going to be an embryo or is it going to be a zygote? Well, we're thinking just the very bare minimum first stage of the process. The moment that the egg and the sperm unite is going to create a zygote. So here is the correct answer. Human intercourse consists of the male introducing sperm into the female's reproductive system. Sperm may then pass through the female's reproductive system to the fallopian tubes, where one sperm fertilizes an ovum, which is the proper term for the female egg is an ovum, and it creates a zygote. The zygote then passes out of the fallopian tube and implants into the uterine wall and begins the gestational period. Over 9 months, that zygote develops and grows into an embryo and then into a fetus. At about 13 weeks, we begin to call it a fetus. It's no longer in the embryonic stage. After that, that fetus is then considered an infant when the baby is born. Okay, so that one's kind of straightforward, but it is kind of tricky when you think about embryo, zygote. I know it kind of confused me at first, but you got to brush up on it, you guys, if that one tricks you up. Next question. Which of the following are included in the male reproductive system? The penis and the epididymis, the vas deferens and the uterus? Is it the penis and the fallopian tubes, or is it the penis, the scrotum, and the cervix? This one right here, it's kind of straightforward, and it is going to be this one here, the penis and the epididymis. Here is what we have, our little diagram, where you can see that the epididymis is definitely part of the male reproductive tract, right? But females do not have this particular anatomy. Right here you have the penis, epididymis. You've also got the vas deferens and the scrotum, testes, seminal vesicles, prostate gland. Definitely brush up on your A&P as it relates to human reproductive anatomy, because you're bound to see some questions asked on there about which belongs where and whatnot. So let's move on to the next question. Which structure plays a role in air conduction? Is it the alveolus, capillaries, lung, or trachea? Now, think here, right here, we have a keyword, structure, right? So automatically start thinking, parts, body parts, not, capillary is not an actual body part. You know what I mean? Like it is, but it's not an actual. I guess it's more like of a thing. Like, it's a blood vessel, but it's not an actual, I don't know. Either way, I hope I'm not confusing anybody, but I'm going to go with the trachea of this right here. And here's why, because the key words right here are going to be air conduction, right? Not gas exchange, but air conduction. So right here, the trachea, it's going to be the primary function of the reproductive system is to provide oxygen to and to remove carbon dioxide from the body. At the air conduction portion, air is delivered to the lungs. This region consists of the upper and lower respiratory tract, which includes specifically the larynx, the trachea, the bronchi or bronchioles. It depends on how you pronounce it. It depends on where you're from, honestly. But the gas exchange portion, that's going to take place between the air and the bloodstream. So it's going to involve smaller parts of our smaller parts of our respiratory tract that are going to include the lungs, the alveoli, and the capillaries. Okay, so if you see gas exchange, you've got to be familiar with the lungs, alveoli, and capillary. Or if we're talking about air conduction, it's going to specifically refer to the structural parts, the larynx, trachea, bronchi, and the bronchioles. Okay, so let's move on to the next question. What body system is the skeletal system most closely associated with when hematopoiesis happens? So here's the keyword right here, hematopoiesis, which is going to be the start of new blood cells and the synthesis of new blood cells, right? So is it going to involve the urinary system, the digestive system, the muscular system, or the cardiovascular system? I'm going to say the cardiovascular system, and here is why. Let's see. Right here, we have the skeletal system consists of bones, cartilage, and ligaments. They're tightly bound together to form a strong yet flexible framework, right? The bone is an active form of connective tissue. So within that bone marrow, there are lots of things going on, you guys. And this tissue plays a role in many functions of the skeletal system. But for this particular question, we're focused on hematopoiesis, right? So hematopoiesis is going to be the process that bones use to produce red blood cells and stem cells, which differentiate to a variety of different cell types in the body. So I'm going to go ahead and go with the cardiovascular system for this one, okay? And why the cardiovascular system is going to be more tied in with the hematopoiesis is because, when you think cardiovascular, you're thinking blood rich, right? So that's going to be the one that's going to be closely associated with the new formation of new blood cells. Obviously, it's going to get out there in circulation. So they're going to be more closely related versus urinary or digestive, which we know are different systems. Urinary dealing with the kidneys and the bladder, digestive dealing with the bowel and, eating and all that. It just doesn't make sense. So right here, the most closely related answer is going to be the cardiovascular system for this one. And we're going to move on to the next question. Which is the final structure through which urine must travel to empty out of the body? Is it the bladder, kidney, ureter, or urethra? So right away, we can eliminate the bladder and the kidney because the key word in this question is going to be the final structure. So I'm going to go ahead and go with urethra because I know that's the body's exit point. And let's see what it says here. Here's a diagram where it shows the urethra. For males, it's going to be at the end. It's this long tube here that resides inside of the penis. For women, it's this super short tube right here, which is going to lead to the outer part of the vagina, or it's going to lead to the outer part of the body where urine comes out. Now, always remember that the female urethra is going to be very short compared to the males, which is why women are at far greater risk of getting urinary tract infections, because an infection doesn't have very far to travel. It can easily go right there to the bladder and infect it, okay? So right here, the kidney's responsibility, if you were thinking kidneys, because the kidney's responsibility is primarily as a filter. It is a filter that excretes urine and excretes waste from the body, including urine, and it's also involved with acid or with the electrolyte balance between sodium and potassium. And we'll talk about that later. All right, question number 17. Which sequence describes the hierarchy level of biological organization? Is it the phylum, kingdom, class, order, family, genus, and species? Genus, class, kingdom, species, order, phylum, and family? Or is it family, species, genus, order, kingdom, class, and phylum? Or is it species, kingdom, genus, class, family, phylum, and order? Well, I'm going to say with this one, because it starts with the word kingdom, I know that's the first one, so I'm going to go with that one. And let's see right here what it says right here. Yep. So the taxonomy is going to be the process of classifying, describing, and naming organisms. So brush up on your taxonomy. If this one kind of stumped you, if you could not remember which one came first, you're going to always start with kingdom, phylum, class, order, family, genus, and species. Right there. It's going to be more specific as you go down. Next question. Which example is a part of the scientific method? Is it a student reads about a new way to harness energy from the sun? Researcher studies the effects of car exhaust on how people breathe? Is it a researcher analyzes how many plants respond well to a new fertilizer? Or is it a student discovers how insulin plays a role in the development of diabetes? I'm going to pick the researcher analyzes how many plants respond well to a new fertilizer. Okay, so right here we have the entire scientific method, which begins with a problem, research, hypothesis, experiment, which is going to include an independent variable and a dependent variable. And they also can include a control group, which is the standard to which you're going to make your comparisons. You're going to observe and come to a conclusion, as well as communicate your findings to others, right? So that they can replicate and verify your results. So with this one right here, this one more closely aligns with the actual scientific method because it gives you a few clues in your answer. One being they're analyzing how many plants, and it's analyzing their response to a new fertilizer. So the rest of these answers, I guess I should say, not questions. The rest of these answers are not quite addressing all of that in their answers. So this one's the more comprehensive, so we're going to go with that one. And we're going to move on to the next question. What standard is used to make comparisons and experiments? We just talked about this one. Is it the sample size? Is it the control group? Is it the dependent variable? Or is it the independent variable? I think we can say that this one's going to be the control group. And the control group is going to be the standard in your study that is going to be used to make comparisons. So, yep, we got that one right. And we're going to move on to the next one. Like I said, the independent variable and a dependent variable, those don't work. But for this one, we can say control group, comparisons. Now you're speaking the language that relates to each other. Control group comparisons, you got CC. So for some people, that might work in your brain. If your brain kind of works a little bit like mine, I'm able to match letters and remember things creatively like that. So let's move on to the next one. Why did it take many years for the cell therapy to be developed? Was it because advancements in microscopy took place slowly? Cells were difficult to isolate for experimental analysis. Researchers believed a cell formed from pre-existing cells, or scientists already proved that cells were essential for life. Well, that's true, but it doesn't address why it took so long. So we can eliminate that one. And right here, I can eliminate researchers believe that a cell formed from pre-existing cells, not so much. And cells were difficult to isolate for experimental analysis. It's tongue-tied. Lots of tongue-tiers, you guys. But advancements in microscopy took place so slowly. So we're going to pick that as our answer. And let's check it and see what it says. Ding, ding, ding. Robert Hooke discovered the first cells in the mid-18th century. The cell theory is a theory because it is supported by a significant number of experimental findings. And the cell theory took many years to be developed because microscopes were not powerful enough to make such observations back then, you guys. It's kind of a no-brainer when you think about the way that, or the advancements that have been made through science. Back in the day, they just didn't have the tools to see cells. So that's definitely going to be the answer for this one. And we're going to move on to the next one. What organelle is only associated with plant cells? Is it the cell wall, ribosome, cytoplasm, or the Golgi apparatus? Well, I can say this takes me way back to my elementary school science classes, and I can say I know that the only organelle that is associated with plant cells is going to be the cell wall. And let's see what our answer key says here. Exactly. There is a plant cell and there is your nice cell wall on the outside. Only plant cells have cell walls, which help protect the cell and provide structural support. Animal cells just don't jive like that. They only have the cell membrane. Okay, so brush up on your science from way back in the day, you guys. If that one kind of tripped you up, definitely revisit the differences between plant and animal cells before you sit for this exam. Next question. Which of the following is supported by the cell theory, which we kind of just talked about in the previous question? Cells are alive and recognized as the building blocks for life. Scientists can identify and differentiate cells by using a microscope. Cells are produced from living cells using meiosis instead of mitosis. And living things are composed of a single cell that remains undifferentiated. Right away, I can say that last one on the list, living cells are composed of a single cell that remains undifferentiated. I can eliminate that. We know living things are typically multicellular organisms, so we can eliminate that one right away. And as far as cells being produced from existing cells using meiosis instead of mitosis, I don't believe that that's true at all. But however, the cell theory refers to the fact that cells are alive and are recognized as the building blocks for life. So if you were stuck between scientists can identify and differentiate cells by using a microscope and this one, that may be true. However, that's not true for this question. That's not what this question is asking. I'll rephrase and say that. That's not what this question is asking. It's asking us to say what is supported by the cell theory, right? And that's just going to be the basic bare bones answer. Cells are alive and they're recognized as the building blocks for life. That's what the cell theory set out to put forth. So you guys, as it relates to the cell theory, after scientists were able to view cells under the microscope, then they were able to formulate the cell theory. And one part of their theory was that cells are alive and they represent the basic unit of life. All living things are composed and made of cells. Cells are the smallest structural units and the basic building blocks of living things. So keep that in mind when you're going over the cell theory. Let's go to the next question, you guys. What raw inorganic material would an autotroph most likely use to create chemical energy for growth? Is it going to be carbon dioxide, minerals in soil, decaying matter, or sugar molecules? I'm going to say we're going to X out immediately sugar molecules. They're going to be way too big for something like this to use, as well as decaying matter. However, two that would probably have me tripped up are going to be carbon dioxide and minerals in soil. But because it's an autotroph, we're going to go with minerals in soil. So let's see. Correct. Autotrophs are organisms that use basic raw materials in nature, like the sun, to make energy-rich biomolecules. Minerals are naturally inorganic, okay? And these are going to be organisms that do this. Of course, they absorb their energy from the sun and the different things that are in the soil. That's what they're going to use. They're going to rely on photosynthesis to transfer the sunlight into food energy that is going to be necessary for their life. So that's going to be the correct answer for this one. And then we're going to move on to question number 24. When would a cell most likely contain the most nucleotides? Is it going to be in the S phase, the G1 phase, the M phase, or the G2 phase? Okay, so let's see. Which one is that going to be? I'm going to go with the G1 phase, and here's why, you guys. The cells are going to copy its DNA during the S phase, and nucleotides are the building blocks of DNA. Thus, the step preceding the S phase is the G1 phase, which is the phase of the cell cycle when the cell would contain the most nucleotides, right? So right here, the G1 is the first gap phase, during which the cell prepares to copy its DNA, followed by the synthesis phase, which is when DNA is copied, and that is eventually followed by the second gap phase, during which the cell prepares for the cell division cycle of mitosis, okay? So moving right along to question number 25. Which is the correct order of the stages of the cell cycle? So please make sure you guys know about the cell cycle and the way these things operate. Is it going to be G1, S, G2, and M? Is it going to be G2, S, G1, or M? Is it going to start with M, S, G2, G1? Or is it going to be S, M, G1, G1? Right away, I can tell you guys, we're going to eliminate all these three. This is just something you're going to have to commit to memory. And right here, we're going to go with G1, S, G2, and M, our first selection on the list. And here is why. The cell cycle is an organized process. It is divided into two phases, which is going to be interphase and the M, or the mitotic phase. During the interphase, the cell grows and copies its DNA. And then once it reaches the M phase, the division into two new cells can occur. But the G1, S, and G2 phases are what comprise of interphase. So the G1 is going to be the very beginning. Always remember that. It's going to be the first gap where the cell prepares by copying its DNA. That's the first step, okay? All right, you guys, next question. Question number 26. A student notices a pattern of stripes on 5 tigers. Each of the 5 tigers has the same stripe pattern. Using his inductive reasoning, what does he logically assume based on this information? The pattern continues to change over time. Natural adaptations cause this pattern to occur. Each offspring will have the same stripe pattern. Or is it that ancestors of the tiger have different stripe patterns? Well, I can tell you right away that this is talking about heredity and genetics, right? And if I'm gonna say that if we can see that we've got 5 stripes on the tigers, or a pattern of stripes on the 5 tigers rather, that particular pattern is gonna be consistent across the board because each offspring will have the same stripe pattern. All right, so here we see the correct answer is each offspring will have the same stripe pattern because Inductive reasoning, not to be confused with deductive reasoning, is going to be where it involves making specific observations and using them to make broad statements, right? The student observes that all the tigers have the same pattern. He can use this observation to make the broad statement that all of the tiger's offspring will have the same stripe pattern. So, within what inductive reasoning is, because that's your keyword in this question, is that it's going to involve drawing a general conclusion from specific observation, making a general statement. So this is referred to as a from the bottom up approach, where information is gathered from specific observations that can then be used to make a general conclusion about the topic under investigation. In other words, you guys, the conclusions are based on observed patterns in data. So basically what this question already tells us that this is coming off of him making a general observation, okay? So you guys, let's move on to our next question, question number 27. A researcher notices a positive correlation between the height of a plant and the nutrient concentration over time. Based on this observation, what conclusion does he reach? The height of the plant increases in the absence and presence of the nutrients. When the amount of the nutrients available to the plant decreases, its height increases. Or is it the amount of nutrients available to a plant is independent of how tall the plant gets? Or last up, is it when the amount of nutrients available to the plant increases, its height also increases? That's a clue right there. We're going to go with this last one on the list, and here is what it says. Because of this positive correlation, if the nutrient concentration increases or decreases, the plant height will either increase or decrease accordingly. So you guys, next question. Which statement best represents Mendel's experiment with garden peas? Was it, as a result, Mendel's developed several theories that have since been disproved? Mendel realized that he was on an incorrect track, which led him to other experimental media. As a result, Mendel developed foundational conclusions that are still valued and followed today. Or was it Mendel collaborated with others interested in genetics to display heredity guidelines that we still use today? However, the correct answer is going to be Mendel collaborated with others interested in genetics to develop heredity guidelines that we still use today. Predicted patterns of heredity. He discovered that plants are going to either self-pollinate or cross-pollinate when the pollen from one plant fertilizes the pistil from another plant. He also discovered that traits are either dominant or recessive, and we still use these tenets today in science and genetics. So, next question. Which of the following is a component of a chromosome? Is it a centromere, gamete, homolog, or ribose? Let's go with centromere, because ribose is going to be referring to a sugar. Homolog is going to be one that is going to refer to something totally not related to the anatomy of a chromosome. And here is a gamete. And gamete's going to be more of along the lines of, I'm thinking reproduction when I hear the word gamete. So let's go with centromere, which we know is actually part of a chromosome. Let's see what our answer says here. There it is. The centromere is going to be in the center of the chromosome that divides the two chromatids. You got one half on top, the other half on the bottom. So the protein-like disc that's going to hold the two sister chromatids together is what collectively makes a chromosome, right? And it's going to have two components. You're going to have the chromatids, which are going to be two copies of each chromosome. And then you're going to have the centromere, which is going to be the protein disc that attach the two chromatids together, okay? Okay, which solution has a pH of 7? Is it aniline, pyridine, pure water, or sodium hydroxide? When you learn about acids and bases, we learn that the pH of 7 is going to represent pure water. So we're going to go with that one. A pH of 7 is a neutral solution, which is how water is classified. Researchers can determine the strength of an acid or a base by measuring the pH of a solution. So we know if it's going to be below 7, it's going to be acidic. If it is above 7, it's going to be alkaline. Okay. Next question. Let's see here. We have question number 31. Which is classified as a type of acid-base reaction that produces a salt? Is it combination, decomposition, hydrolysis, or neutralization? We're going to go with neutralization for this one, and here is our rationale. A neutralization reaction is a type of acid-base reaction where an acid and base react together to form a salt and water. So with this, in an aqueous solution, a base increases the hydroxide concentration, while an acid increases the hydrogen ion concentration, right? And because of that, sometimes neutralization can occur. This type of reaction happens when an acid and a base react with each other to form water and salt. And salt is typically defined as an ionic compound that includes any cation except hydrogen and any ion except for hydroxide. So let's see here. We're going to move on to our next question. Question number 32 is, which of the following determines the strength of an acidic solution? Is it litmus paper that turns red, litmus paper that turns blue, measured pH value equal to 7, or a measured pH value less than 7? So we just kind of went over this a little bit ago when we were talking about the neutralization, and we're going to say any pH that is measured less than 7 is going to be considered acidic. Let's see if we're right. Correct. Both litmus paper and a pH scale can be used to indicate whether a solution is acidic. However, a pH scale can also determine the strength of the acid. That's the key right there. The strength of the acid. Okay. Can be far more, Be far more specific when you have the actual pH strips. Next question. Which type of bond forms between nitrogen and oxygen and why? Is it ionic because electrons are shared? Is it covalent because electrons are shared? Is it ionic because electrons are transferred? Or is it covalent because electrons are transferred? Well, you guys, for this one, we're going to go with covalent because by definition, covalent indicates that there's going to be a sharing of electrons. And here we have it. So right here, nitrogen and hydrogen are both nonmetals, which means they will share electrons in a covalent bond. For example, 2 oxygen atoms form a double bond, which are 2 pairs of electrons, and you have 4 total electrons, and then you have right here 2 nitrogen atoms that form a molecule are going to form a triple bond in which 3 pairs of electrons in total are shared. So here are your covalent bonds right here that are represented by the dots or by the 2 lines. That means they're sharing right here. And let's move on to the next question, which is going to be, which of the following atoms is a cation? Is it going to be 14 protons of 14 neutrons and 18 electrons? 34 protons, 45 neutrons, or 36 electrons? Is it going to be 35 protons, 44 neutrons, and 35 electrons? Or is it going to be 82 protons, 125 neutrons, and 78 neutrons? We are going to go with the last one on the list because it's going to be the one that has the most protons between protons and electrons. We're going to go with 82 because that's more than 78. For this particular instance, we're going to ignore this category of neutrons and focus specifically on what is the more positive charge. Protons are going to give that atom a positive charge, whereas electrons will give it a negative charge. That's where you're going to end up with anions. But in this case, we're talking about cations. So we're going to go with 82 125 neutrons, and 78 electrons. And this one is correct because it has more protons than electrons, and this atom has a positive charge, which is what I was saying, and can be classified as a cation. When a metal such as sodium reacts to become stable, it loses its valence electrons. And at first, it is a neutral atom with 11 protons and 11 electrons. However, when it loses an electron, the number of protons does not change. It does not change. And the atom has 11 protons and 10 electrons. Because there is one more positively charged proton, a cation forms. And a cation is an ion that has a net positive charge, okay? Try not to confuse that with anion. Definitely make sure you understand the difference between an anion and a cation. Next question. A spoonful of sugar is added to a cup of hot tea. All the sugar dissolves. How can the resulting solution be described? Is it saturated and homogeneous, saturated and heterogeneous, unsaturated and homogeneous, or unsaturated and heterogeneous? Answer is going to be unsaturated and homogeneous, and here is why. And that's going to be because more solute can be added and dissolved. Just because you added a teaspoon of sugar doesn't mean, or a spoonful of sugar doesn't mean that the whole entire mixture is saturated with sugar in the mixture. So right here we have that more could be added and dissolved, and the solution has not yet reached its limit and is considered unsaturated. And because all the solute dissolves, the particles in the mix are evenly distributed as a homogeneous mixture. So that's going to be our answer for him. A homogeneous mixture is when substances mix evenly, and it is impossible, to see individual components, whereas a heterogeneous mixture is when the substances mix unevenly, and it is possible to see individual components. So you think a salad, a fruit salad, you can always pick out the grapes versus the pineapples versus the cantaloupe, the apples, the blueberries. You can pick them out individually. However, with the homogeneous, you can't pick out individual sugar molecules and take them out and say, here's my sugar. It's just not going to work. So definitely make sure you understand the different properties and states of mixtures and matter and all that stuff too. You got to brush up on those things before you try to sit for the TEAS exam. Our next question is going to be, this is question number 36. In the following single replacement reaction, blank replaces blank. Is it the sodium and the iodine, chlorine, iodine, chlorine and sodium, or sodium, chlorine? So the answer is going to be the chlorine and iodine, and here is why. In this reaction, the chlorine is an element in the reaction that replaces the iodine in the compound sodium iodide. This allows chlorine to form a compound with sodium as it leaves iodine as an element. Okay, so these two combined, but this one was left on its own. Okay, so there's your answer. The answer is going to be that the chlorine replaces the iodine. What type of reaction is described by the following equation? Is it a synthesis, decomposition, single replacement, or is this a double replacement reaction, you guys? So you got to look at this and use this as your dividing factor. Right here, this arrow, what's happening over here versus what happened over here on the right side of our equation. So the answer for this one is going to be double replacement. And we're going to take a look at this one right now to see what's the rationale behind this. So in this particular reaction, or particular equation, I'll say, in this reaction, we have two elements that are trading places. In the reactants, you have zinc and bromide ions that are together, and you have potassium and hydroxide ions that are together. However, in the product of this equation, you end up with zinc and hydroxide ions that are together, as well as the potassium and the bromide ions are together. So this is going to be a double replacement reaction here. Next question. Blank is dependent not only on temperature, but also on the amount of substance available. Is it condensation, deposition, evaporation, or is it melting? Right here, we're going to go with evaporation, and here is why. Unlike condensation, deposition, and melting, evaporation is dependent not only on the temperature, but also on the amount of substance available, okay? So right here, you've got condensation that's, turning things from a gas to a vapor of liquid. And then with evaporation, you have the change of a liquid to a gas. So how fast and whatnot that happens with evaporation is definitely going to be dependent on the temperature, as well as the amount of the substance. Alrighty, for the next one, during which of the following phase changes will the cohesion between the particles in a substance decrease? When we say cohesion, we're talking about its stickiness factor, the ability of the molecules or atoms to stick together like this, right? Is it going to be condensation, deposition, freezing, or vaporization? Now, keep in mind, it's going to be the phase where the cohesion is going to be decreasing, going down. I'm guessing vaporization. Let's see if we're correct here. Ta-da! So if the cohesion between particles decreases, meaning it's not held together as tightly, then the particles must be undergoing a phase change that will allow the particles to move farther apart. And this happens when a substance vaporizes and turns from a liquid into a gas. So right here, you can see how this happens right here. There you go. And our next question. In which state of matter are the intermolecular forces between particles in a substance the strongest? Is it gas, liquid, plasma, or solid? So you guys, our keywords here are going to be, when are the particles in the substance the strongest? When are they held together the tightest? My little brain and my little antennas are up, and I'm saying solid, you guys. What do you say? We're going to go with solid. Let's see what it says for the answer key. In solids, particles are usually closer together than in other states of matter because of the strong cohesive forces between the particles. So here you have that solid. They're as tight as can be, you guys. Okay, so that one was kind of an easy one. But just remember, solid's going to be the strongest in your states of matter. And the next question. Mendel discovered the pattern associated with blank after developing a series of rules in genetics. Is it epigenetics, heredity, heterogeneity, or taxonomy? Mendel was dealing with patterns in heredity. Okay, so we're going to go ahead and pick that one. And let's see what our answer check says. Correct. Mendel was accurately able to predict the patterns of heredity by studying the rules related to genetics. These rules help shape his theory of heredity, whereas heredity refers to the characteristics that offspring inherit from their parents. Next question. The sequence of amino acids in a gene determines primary structure of a codon, the primary structure of a protein. Is it the primary structure of a nucleotide, or is it the primary structure of a nucleic acid? The sequence of amino acids are going to be the building blocks of proteins. So, when I hear the word protein, I'm automatically also thinking about amino acids, which make up the larger chain of proteins. So let's see what we have right here. Let's check our answer. And right here, the sequence of amino acids in a gene determines the primary structure of the protein. The components necessary for translation are located in the cytoplasm. Basically, the components that bring all this together is going to all deal with proteins. Sequence of amino acids determines the protein structure, which then determines its function. Okay. Next question. Which is true regarding the urinary system? Select all that apply. So yes, there's gonna be some of these on your test, so hopefully not too many. So is it protein or the kidneys make urine? As a person ages, kidney tissue and filtration capacity increase? Is the kidneys help regulate water balance? Or is it that they regulate the levels of electrolytes such as sodium and potassium? Or is it eliminates metabolic waste? Now, these are all the functions of the urinary system. Okay? For this particular question, we're going to go with the last three, and here is why. So if you were thinking that the kidneys make urine is correct, that's not correct. That's totally incorrect. They do not make urine. However, they filter and they help to regulate water balance as well as regulating the levels of electrolytes such as sodium and potassium as well as eliminating metabolic waste. Urine is going to be the byproduct of these functions. And also, as a person ages, their kidney function and their kidney tissue and filtration capacity are going to decrease, okay? So that was incorrect. However, kidneys help regulate water balance, regulate the level of electrolytes, as well as eliminating metabolic waste. Those are all things that are functions of the urinary system. Okay, next question. During the aging process, not all hormone levels decrease. Some actually increase. Which of the following is a hormone that may increase as a person ages? Is it going to be cortisol, insulin, luteinizing, or thyroid? Which one of these hormones, you guys? What are you thinking? I'm going to go with luteinizing for this one. I'm going to tell you why. So with this right here, the aging process, you've got multiple different hormones that go through changes throughout the aging process. But hormones that are going to be decreasing as the body ages, for women, it's going to be estrogen. For men, it's going to be testosterone. Growth hormone also decreases, as well as melatonin, which helps keep people asleep or helps people to get good sleep or whatnot. Now, in women, we also have, or within the body, we also have different hormones that remain unchanged or decrease slightly. And those include cortisol, insulin, and thyroid hormones. However, you have some that are going to increase as you age, and that's going to be the follicle stimulating hormone, or FSH, the luteinizing hormone, or LH, norepinephrine, epinephrine in the very old, or the parathyroid hormone, you guys. So definitely check up and brush up on your hormones that are common in the body. You definitely need to know that stuff. Question number 45. What phase is the cell cycle a part of? Is it interphase, metaphase, prophase, or telophase? The cell cycle, when you have the gap 1, gap 2, you have the S phase, that's going to be part of the interphase cycle of the cell. And here we have it right here. Growth, we have growth 1, you have just G1 rather, growth, DNA replication or your synthesis phase. Then you're preparing for division, which is your gap 2 phase, and there's that. So interphase is going to be all these three with the exception of that. And we're moving right along to the next question. Question number 46. In which state of matter do the particles of iron have the lowest amount of cohesion? Is it solid iron particles have the lowest amount of cohesion? Liquid iron particles that have lowest amount of cohesion? Is it gaseous iron particles that have the lowest amount of cohesion? Or is it the particles have the same amount of cohesion in all states of matter? Which one, you guys? I'm going to go with gas because gas are going to be scattered about. I can imagine gas particles are scattered about in the air. We know that here's the answer. Gaseous iron particles have the lowest amount of cohesion. A person is diagnosed as having acidosis, a condition in which the blood pH is below 7.5. What does the doctor most likely conclude? Is it that there's too much carbon dioxide found in the blood? Is it highly oxygenated blood circulates through the body? Is it a blockage prevents blood from leaving the pulmonary artery? Or is it the nasal cavity has a difficult time clearing particles from the air? Which one is it, you guys? We're going to go with too much carbon dioxide is found in the blood right here. And here is why. Acidosis is when the body fluids contain too much acid or too low of a pH. The kidneys and lungs are unable to keep the body's pH in balance. Acidosis is the result of when there is too much loss of the bicarbonate from the blood, known as metabolic acidosis, or as a buildup of carbon dioxide in the blood due to poor lung function, which is also known as respiratory acidosis. It is the opposite of alkalosis, which is a condition in which there is too much base in the body fluids. Next question, question number 48. An atom has 28 protons, 32 neutrons, and 28 electrons. What is the name of this isotope? Is it nickel 32, nickel 60, germanium 56, or is it germanium 60? Well, there's some adding here going on, so we're going to go ahead and add the protons and the neutrons together to come up with nickel 60. And let's see what it says here. So the number of, it's correct. So the number of protons, 28, is going to give us our atomic number, right? Which is going to be on the periodic table. And that's going to identify this atom as nickel. And the mass is the number after the dash in the isotope name, which is determined by adding the numbers of protons and neutrons together, which is going to be 28 plus 32 is going to equal 60. So we're going to pick nickel 60 for this one. Question number 49. The physical appearance or blank of an organism is determined by a set of alleles. Is it genotype, phenotype, transcription, or translation? When you hear the word physical appearance, you're going to want to definitely go with phenotype. These are two peas that are together like a pea, two peas in a pod, literally. Physical means phenotype, right? So let's go right here, click check. And a phenotype is the physical appearance of an organism, and the genotype is the actual set of the alleles, okay? So the set of alleles, or the set of genes, is going to determine the outward physical appearance that we can see, which is the phenotype. And last but not least, question number 50, you guys. It's a tricky one, so be prepared for these. Remember what I said about learning your AMP as far as the gastrointestinal tract is concerned? Here's why. Question number 50 is arrange the path of food through the digestive system. Move the steps into the boxes below, placing them in the correct sequential order. Use all of the steps. So think about what happens when you take a bite of a cracker. It's going to be in your oral cavity first of all, but what happens after it leaves the mouth? So we're going to go ahead, we're going to start with what we know, which is going to be the oral cavity. It's going to be the first part, that's the mouth. We're going to go from oral cavity, right? Think of that diagram where you see the guy and it's like a side profile and you can see the mouth and it goes down. It's going to start in the oral cavity. Then from there, it's going to go to the throat, the pharynx. Then from there, it goes to the esophagus. And from there, we go to the pyloric sphincter, down to remember earlier, I said it was the small intestine where all your nutrient absorption is going to take place. And it eventually goes to the large intestine. And from there, we go to the colon before it exits the body via the rectum and anus. Now, let's check our answer and see what it says right here. Ta-da! We've got it. There it is. You've got the oral cavity, which is the first part of the digestive system. It is bounded by the lips and cheeks and contains the teeth and tongue, and its primary function is to masticate or chew and moisten the food with the saliva. That saliva, it begins the digestive process. The pharynx or the throat connects the mouth to the esophagus, and the esophagus is a muscular tube that's about 25 centimeters long. Food travels down, I consider it going down the hatch, and it goes to the, it says right here cardiac sphincter, but that is incorrect. It is the pyloric sphincter, which is right here. The pyloric sphincter comes next in the list, and that is the exit of the stomach, and it connects, yes, the pyloric sphincter connects the small intestine, not the esophagus and the stomach, so I gotta brush up on my own. But yes, you guys, the small intestine has the pyloric sphincter right here that keeps things going one way. We don't want things going backwards. If it goes backwards from the small intestine on up to the esophagus, you're in trouble. Okay? Point blank, you're in trouble. From there, it's going to go to the large intestine. Let's go back up. The small intestine is about 6 meters, and it consists of 3 parts: the duodenum, jejunum, and the ileum. And from there, your large intestine contains the cecum, the colon, the rectum, and the anal canal. And the cecum is located exactly where the small and large intestine meet, okay? And your colon right here is at the very end. It's going to be about 1.5 to 1.8 centimeters and consists of 4 parts, which is going to be the ascending, the transverse, the descending. Wait, it's going to be right here. Sorry, guys. Ascending, transverse, descending, going down right to the sigmoid colon, which is right around here, right on out to the rectum, and this right here exits the body. I want to thank you guys. You did a great job following along with this test. Please be sure to check the link in the description box for your entire free T7 practice test, you guys. You cannot beat that offer right there. Definitely take it. Brush up on your skills if you need to, and don't forget that you can join the online course as well as the TEAS study group.